Hello, everyone. I'm Kennedy Dorsey. I am a seventh grade science teacher in Maryland. In today's lesson, I will present on E. coli O157 H7. This is a significant foodborne pathogen that is responsible for causing hemorrhagic colitis and hemolytic uremic syndrome, also known as HUS. The reason for me choosing this organism is due to the fact that it highlights the cross between microbiology, human health, and public safety. Normally harmless strands of E. coli live in our intestines. However, this particular strain is highly pathogenic and can cause severe illness, even at very low doses. In this presentation, we'll go through the classification, structure, and how it causes disease. In addition, this presentation will go over how the outbreak occurs and the current approaches to treatment, prevention, and public health. Lastly, this presentation will share examples of pathogen is documented outbreaks that show why this pathogen is closely monitored by different agencies like the CDC, WHO, and the FDA. My goals for this presentation is to not only explain the science, but to also give you a clear understanding of why this bacterium remains such a concern in food safety worldwide, and also prevention efforts are critical. So take a look at these objectives on the screen. Before we begin, I'll give you a quick overview of what will be covered in this lesson. We will first talk about how E. coli O157 H7 differs from the normal strand that lives in our gut. Although most strands of E. coli are harmless and even helpful, this particular strand has special features that make it very dangerous. We will also take a look at how this organism actually makes people sick, the process called pathogenesis. This will also include the toxin that it produces and how it damages the intestines and why it can lead to serious complications. This most likely leads to serious complications like kidney failure and hemolytic uremic syndrome or HUS. In addition, this lesson will explain how bacteria spreads. You will learn that it does not take much. Very few bacteria can make someone sick. We will look at the common source like undercooked beef, raw vegetables, and contaminated water. We will also figure out why outbreaks continuously happen. Lastly, we will connect real-life outbreaks to the public health actions that follow. For example, food safety regulations have changed a lot due to this bacteria, and research has looked into vaccines for cattle to be able to reduce the exposure. So, who am I, or what am I? So, I am Escherichia coli O157 H7. I am the causative agent of two serious illnesses, hemorrhagic colitis and hemolytic uremic syndrome. As stated above, many of the E. coli strains are harmless members of the human gut microbiota. The strain is part of the enterohemorrhagic E. coli group, which produces Shiga toxins. These toxins can damage intestinal cells and blood vessels. Unlike conventional E. coli that aids in digestion and vitamin K production, E. coli O157 H7 is a pathogenic and can cause severe disease even with low infection dose. As few as 10 to 100 organisms can lead to illnesses. Historically, this pathogen has been associated with high-profile outbreaks that are linked to undercooked ground beef, contaminated fresh produce such as spinach and lettuce, in addition, unpasteurized juices, and even recreational water sources. In 1993, a famous outbreak occurred in the United States, causing hundreds of illnesses and several child deaths, drawing national attention to food pathogens. We'll take a quick pause to turn and think about the question. Why do you What are some clear distinctions between harmless gut E. coli? With a clear understanding, it helps others see why public health agencies treat agencies with creating a sense of urgency as it relates to this topic. The history behind E. coli starts with it being recognized as a serious human pathogen in 1982. In that year, there were 2 outbreaks of severe bloody diarrhea. This was traced back to contaminated hamburgers that were served in a fast food restaurant in Oregon and Michigan. This investigation was led by the Centers for Disease Control and Prevention, also known as the CDC, that concluded that this was for the first time E. coli O157 H7. This marked as a distinct pathogen strand against the typical strand that was found in the gut. One thing that made this discovery significant was not only the unusual severity of this disease, but also the link to foodborne transmission. The CDC's epidemiological work demonstrated that the power of outbreak investigations and foodborne illness tracking in identifying new pathogens. This was done by analyzing patients' stool samples, food sources, and epidemiological patterns. This will confirm that this was a previously unrecognized cause of humoral colitis. With further research, it was revealed that the strain produced potent Shiga toxins. They were named this because they are structurally and functionally related to the toxin that's produced by Shigella dysentery. This toxin damaged intestinal endothelial cells, leading to bloody diarrhea, and in some cases triggering systemic effects that cause hemolytic uremia syndrome. Due to the ability of E. coli and O157 and the related strands now are classified as Shiga toxin-producing E. coli or STEC. Identifying this pathogen in the 1980s marked a turning point in food safety. It brought attention to the risk posed by undercooked beef, unpasteurized products, and cross-contamination in food preparation. Since the 1980s, regulatory standards, surveillance systems, and public health significantly detect and control STEC outbreaks. Now let's take a look at the taxonomy triangle. So at the top of the triangle, we have where E. coli O157 has a domain called bacteria. The phylum is known as the Protobacteria. Their class is Gamma Proteobacteria. Their order is Enterobacteriales. Their family is Enterobacteriaceae, and their gene or species is known as Escherichia coli. And lastly, their stereotype is known as O157 H7. Okay? And again, I'm going to keep repeating to let you guys know, although that most strands are harmless of the human intestinal tract, this particular serotype, O157H7, is associated with severe gastrointestinal disease and systematic complications. The designation O157, the H7 refers specifically to the bacterium serotype, which is determined by the antigenic properties of its surface structures. In this serotype, the O refers to the antigen, a part of the lipopolysaccharide found on the outer membrane of gram-negative bacteria. In this case, O157 indicates the 157th identified variation of the somatic O antigen. Variations in this O antigen play an essential role in immune recognition and pathogenic potential. The H antigen refers to the flagellar protein that provides motility. The H7 designation indicates the seventh identified variation of the flagellar antigen. The flagellar antigen not only aids in motility, but also contributes to immune system interaction and bacterial virulence. By combining both the O and the H antigens, microbiologists can classify E. coli strands into over 1,880 recognized serotypes. Among them, the O157 is the most notorious due to its association with the hemorrhagic colitis and the hemolytic uremic syndrome. Having an understanding of the serotyping system is important for public health surveillance because it allows rapid identification of pathogenic strands during outbreaks. This distinguishes them from Harlem's commissal E. coli. Now, if you look at the structure, we have the bacteria. Now, around it is labeled with the different parts. So E. coli is a gram-negative bacillus, meaning that it has a rod-shaped morphology in a cell envelope that is characterized by an outer membrane with lipopolysaccharide, a thin peptidoglycan layer, and an internal cytoplasmic membrane. In the outer membrane, the lipopolysaccharide contains the O antigen, which is the major determinant in serotyping and an important component in immune system recognition. The bacteria is able to move due to being surrounded by flagella, with the H7 antigen representing a specific structural type of the flagellin protein. Motility not only helps the bacteria reach and colonize specific areas of the gastrointestinal, but also contributes to immune recognition and in some cases virulence. The highlight of this bacteria is its ability to produce Shiga toxins. They are AB type exotoxins that inhibit the protein synthesis by targeting the 29S ribosome RNA of the ribosome, leading to the host cell's death. Both of the toxins can cause intestinal damage. It is strongly associated with severe outcomes like hemolytic uremic syndrome. These toxins are encoded by bacteriophages integrated into the bacterial genome, making them powerful and mobile virulence factors. In addition to toxins, adherence mechanisms are essential. E. coli O157 H7 possesses fimbriae, also known as pili, that help mediate the initial attachment to the intestinal mucosa. These structures, along with other adhesions, allow the bacteria to establish contact with epithelial cells. Once attached, the bacterium deploys a type 3 secretion system encoded on the locus of endocyte and effacement pathogenicity island. The system injects bacterial effector proteins directly into the host cells, leading to attaching and effacing lesions characterized by loss of microvilli and actin pedestal formation. Now let's look at how it causes disease. So it causes disease by first exposure. The infection is followed by the ingestion of contaminated food or water, so such as undercooked beef, leafy greens, unpasteurized juices. Escherichia coli has acid-resistant systems that allow it to survive gastric acidity, so as few as 10 to 100 organisms can cause disease. After incubation of about 3 to 4 days, the patients begin to develop abdominal cramps and watery diarrhea. The diarrhea may progress to bloody diarrhea. Secondly, colonization and attaching and effacing lesions in the lower smaller intestine and colon. Bacteria uses adhesions and fimbriae to stick to the intestinal lining. The LEE pathogenicity island carries instructions for a type 3 secretion system, which acts like a syringe to inject proteins into intestinal cells. The bacterial sulfurs protein intimate then attaches to the tear, allowing the bacteria to tightly attach and form attaching and effacing lesions. These lesions damage the microvilli, leading to an actin pedestal formation and loss of normal surface structures. When the microvilli destroy nutrient absorption and the protective barrier are disrupted, this explains why the cramping abdominal pain shifts from watery to bloody diarrhea. This is due to intestinal lining becoming more severe. So let's look at the clinical manifestation. So E. coli is particularly dangerous for high-risk groups such as children under 5 years of age, elderly adults, and those who are immunocompromised. These two major outcomes of the bacteria is called hemorrhagic colitis and hemolytic uremic syndrome. Hemorrhagic colitis is a condition where patients often begin with severe abdominal cramps and watery diarrhea, as the bacteria damages the intestinal lining through attaching and effacing lesions and the Shiga toxin production. The diarrhea turns bloody. Unlike many other bacterial infections, patients usually do not develop a high fever, which can help distinguish this illness from others. Now, the hemolytic uremic syndrome is the most dangerous complication caused by E. coli. It is especially dangerous in young children and the elderly. The toxin can enter through the bloodstream and damage the blood vessels. This leads to hemolytic anemia, which is where red blood cells are destroyed. It also leads to thrombocytopenia, biocytopenia. This is where platelets are used up or destroyed, increasing the risk of bleeding. Lastly, acute kidney failure can occur because small blood vessels in the kidneys are particularly vulnerable. The reason why children are the most vulnerable is due to the fact that their kidneys are still developing, making them more susceptible to toxin damage. Even a small amount of the toxin can trigger pus in younger patients, which is why outbreaks linked to food often result in pediatric hospitalization. So, E. coli spreads easily with a very low infection dose. It spreads based upon geographical distribution. Many large-scale outbreaks have occurred in the United States, Canada, and several European countries and Japan. This shows that the pathogen is not just limited to one region, but it is a global public health concern. This pathogen is spread through the most common source, which is undercooked ground beef. This can happen due to the fact that the bacteria being mixed in throughout during processing. Other common vehicles include unpasteurized milk, juice, and fresh produce that is irrigated or washed with contaminated water. Additionally, direct contact with animals in a petting zoo or farm has caused outbreaks in children. The pathogen spreads through the fecal-oral route. Most people ingest the bacteria when food, water, or surfaces are contaminated with tiny amounts of fecal matter. It is important to know where there is an extremely low infection dose that is fewer than 100 bacterial cells. This is enough to cause a disease. His explanation on why the outbreak spreads so quickly and affects large numbers of people. Even minimal cross-contamination during food preparation can be dangerous. This infection tends to peak in the summer months. This is likely due to outdoor cooking and barbecues. An increased consumption of fresh produce. Warm temperatures also support bacterial growth in food if refrigeration and handling are not ideal. Lastly, children are a part of the high-risk group due to the exposure at places where hand hygiene is inconsistent. In the treatment of E. coli O157, the main approach is supportive care. This means that patients are given IV fluids or oral hydration to replace the lost water and electrolytes from diarrhea. This helps to prevent dehydration, which is a major cause of complications. Generally, antibiotics should be avoided. This is really important as E. coli infections. Antibiotics can cause stress bacteria to release even more toxins, worsening the patient's condition. This is often an exam or discussion question. In most severe cases, especially in children, hemolytic uremic syndrome, patients may require dialysis or blood transfusions. It is important to also emphasize that there is no specific cure treatment is more about supporting the body while it recovers. Due to the fact that treatment options are limited, prevention is key. It is important to cook beef to the proper temperature. It must reach 160 degrees internally because bacteria can be mixed throughout. Pasteurization of milk and juices kill pathogens that may be present in raw products. Many outbreaks are linked to unpasteurized drinks, hand washing after handling animals, petting zoos visits, or raw food is critical especially for children. In addition, surveillance systems like those that are run by the CDC in the United States and who internationally help to detect these outbreaks quickly and trace them back to the original source and prevent further spread. Lastly, public education such as campaigns should be remind people about safe cooking and cross-contamination and prevention and hygiene practices. So this is some public health implications of particular pathogen. So E. coli holds an economic, social, and global effect. One of the biggest challenges is the complexity of the food supply chain. Contamination can happen at multiple stages, from the farm to the slaughtering to the meat processing, and even through packaging facilities. In addition, the contamination can happen during transportation and storage. As stated, E. coli has a very low infection dose. Even tiny amounts of contamination can lead to large outbreaks. This makes food supply safety an ongoing challenge. Secondly, this pathogen creates a heavy financial burden. Medical costs from treating severe causes, including dialysis and long-term care for kidney damage. Recalls on products can cost companies millions of dollars and damage public trust. Lastly, Lawsuits and settlements such as the Jack in the Box outbreak can hold parties responsible. So it is very important that we focus on farm-to-table interventions as it gives us better livestock management and improved sanitation and vaccination trials. Later in this section, we'll talk about the future direction of E. coli and how it relates to prevention, treating, and the complications that arise from it. So, future direction. So, rapid diagnosis tests have been advanced from the PCR test, and this helps to clinically diagnose cases earlier, initiate supportive care, and alert public health agencies to potential outbreak. The microbiome and the prebiotic research helps to explore the role of the gut microbiomes. It strengthens the protective role of the natural microbiome. It may be possible to reduce colonization, toxin release, and progression to severe diseases such as HUS. Future directions can be integrated into international surveillance systems. Low and middle income countries will especially benefit from affordable diagnostic and data sharing initiatives supported by the WHO. In conclusion, as we conclude this presentation, let's tie it all together. We talked about Escherichia coli, which is a pathogen that causes severe illness, especially in children and the elderly. Due to the fact that antibiotics are not recommended, there is no specific cure. Treatment is limited to supportive care. This highlights why prevention is the cornerstone of control. Preventing outbreaks requires a multidisciplinary approach. Research helps us understand the pathogen's mechanisms, which public health policy and food safety regulations ensure that this science is applied in the real world. The outbreak in 1993 from Jack in the Box shows how failures in food safety can ripple into legal, economic, global, and health consequences. For the future, emerging tools such as rapid PCR diagnostics, whole genome sequencing, and microbiome-based strategies are shaping the future of outbreak prevention and response. However, technology alone isn't enough. Public education and food safety and global cooperation across supply chains will remain critical. After watching this presentation, I want you guys to ask yourselves or answer this question. What is the greatest challenge in preventing E. coli 0157 H7? Also, what is the greatest outbreak regulation in food industry that we can change the consumer's behavior or overcoming scientific limitation in treatment and detection? Again, I want to say thank you for watching my post. Here are my references. I hope you guys have a good day.